Yes, Michael, and good morning, everyone. So today's lesson we continue with sexual reproduction in flowering plants. Okay, so we are going to talk about the formation of gametes in flowering plants as well as the double fertilization process. Okay, so now first things first, let's get into microgametogenesis. Okay, so microgametogenesis is the development of the pollen grain. Pollen grain, also known as the male gametophyte. Okay, so here you can see. Okay, this picture here show you how this male gametophyte look like, right? Okay, now this microgametogenesis occurs on the anther. Okay, anther of the male plant. Some plants they have both male and female reproductive system in one plant. All right. So now on the anther, the spore mother cells, right? The spore mother cells are also known as the diploid microsporocyte. Okay, you can see from here. These are the microsporocytes, all right? Diploid microsporocytes, okay, which can be found in each pollen sac. This pollen sac also known as the microsporangium. So this is the microsporangium, all right? This structure here, right? So in one anther we have 4 microsporangium, right? So inside this microsporangium we can see these microsporocytes, right? So these microsporocytes, right, will undergo meiosis 1 and 2 to form a um, microspore, right? So now one microsporocytes, okay, undergoes meiosis 1 and 2, okay, to form 4 microspores. And these microspores, right, apparently they are haploids, okay, because you know that meiosis will give a haploid cell or a haploid outcome from a diploid cell, okay. So next, this microspore, what happened to this haploid microspore? Okay, please recall there are 4 haploid microspore, right? So each one of these haploid microspore will undergo mitosis to form mature pollen grain. Okay, and this mature pollen grain, also known as the male gametophyte. And this mature pollen grain or male gametophyte, right? It has, okay, a tube cell. You can see the tube cell here. This is the tube cell. All right, this tube cell has tube nucleus. All right, and okay, this mature pollen grain has a generative cell. So I'm meaning to say one mature pollen grain has tube cell and generative cell in it. Okay, like 2 in 1. Okay, 2 cells they combine together to form one big cell, which is the mature pollen grain. Now, let's take a look at the megagametogenesis, okay, which is the development of a megaspore, okay, into an embryo sac. This embryo sac also known as the female gametophyte, and this megagametogenesis occurs in the ovule, right? So you can see this is the couple, stigma still couple, and then you can see the ovule here, right, inside the ovary. One ovary has many ovules, right? So inside this ovule, right, each of these ovule they are megasporangium, diploid megasporangium. So as couple, okay, couple, okay, develops inside the megasporangium of each ovule, the diploid megasporocytes, okay, will divide by meiosis 1 and 2, which eventually produce 4 haploid megaspore. Okay, so you can see there are 4, 1, 2, 3, 4, 4 haploid megaspores. The outcome of meiosis, okay, from the megasporocytes. Okay, now let me rephrase that. This is the megasporangium, right, which can be found inside the ovule. This megasporangium, right, has megasporocytes. Now, the megasporocyte, this is the megasporocyte. Okay, megasporocyte inside the megasporangium. This megasporocyte divides by meiosis 1 and 2, okay, to form 4 haploid megaspore. All right. Megasporocyte, also known as megaspore mother cell. Okay, so now we have 4 haploid megaspore. So out of these 4 megaspore, only 1 megaspore will survive and, okay, develop. Eventually, it will give rise to an embryo sac, which is the female gametophyte. Okay, now the remaining 3, like in this picture here, diagram here, there are other 3, right? Megaspore. Okay, these other 3 megaspore will degenerate, and the embryo sac cell, right, which was developed from the surviving megaspore, right, this embryo sac cell will divide 3 times by mitosis to form an 8 nuclei cell. All right, these are the 8 nuclei cell which later develop into a matured embryo sac. Okay, so meaning to say that the embryo sac, right, will develop into a matured embryo sac, and this matured embryo sac has 8 nuclei. Okay, so you can see here the 8 nuclei, they are the 3 antipodal cells. Okay, 1 2 3, right, and then it has 2 polar nuclei. You can see the polar nuclei in the central cell, and then it has 2 synergid cells. You can see the 2 synergid cells here. Okay, and an egg, a haploid egg, okay, that is being squeezed between the 2 synergid cell. Now the ovule, okay, the ovule, what will happen to this ovule? If the seed is fertilized, the ovule will become a seed. Okay, and then this ovule, right, now consists of the embryo sac, right, which is enclosed by the megasporangium and 2 surrounding intergumens. These are the 2 surrounding intergumens. 1 2. Okay, surrounding means these 2 intergumen, it surrounds this embryo sac, right? You don't count it as 1 2 3 4. No, there are only 2 intergumens that surround this embryo sac, right? Okay, now we proceed with double fertilization. All right, I'm sure you have learned this in Form 5. All right, now this is more detail. Okay, so this double fertilization is a process in angiosperm. Angiosperm means flowering plants, plants that produce flowers. Right, it's a process in angiosperms that involves the union of different nuclei of the female gametophyte. And you know, a female gametophyte or the mature embryo sac has 8 nuclei, right? So it's the union of these different nuclei of this female gametophyte with 2 male gametes or sperm, right, from the male plant. So this double fertilization starts with a pollen grain. You can see this diagram here. This pollen grain, it adheres to the stigma of the carpel, right? Now this carpel is the female reproductive structure of the flower. Next, the pollen grain, all right, will takes in moisture and begins to germinate and form a pollen tube. You can see the pollen tube here, right, that form from the pollen grain. This pollen tube will extend down toward the ovary, right, through the style. This is style, stigma, style. Now the tip of the pollen tube then enters the ovary. You can see here the tip of this pollen tube enters the ovary and penetrates through the micropyle opening in the ovule. Okay, next, this pollen tube, okay, we proceed to release 2 sperms, all right, 2 sperms into the matured embryo sac. Okay, let me refresh that. This is the embryo sac, right, matured embryo sac. Okay, next, 1 sperm will fertilize the egg cell here, right, and this fertilized egg cell will form the zygote, while the other sperm will fuse with the other two polar nuclei, okay, of the large central cell, and this eventually forms a triploid nucleus. Okay, now the cube nucleus will degenerate, okay, the other synergids, okay, degenerate, right, and the antipodal cells also degenerate. Atau senang cerita hilang bye-bye right. Okay, so now what's left is the matured embryo sac, and then we have the triploid nucleus, which will develop into endosperm, and then the zygote, right. Now, this double fertilization ensures that the endosperm, okay, develops only in ovules where the egg has been fertilized. So, preventing angiosperms from squandering nutrients on infertile ovules. Okay, ini antara kepentingan double fertilization. Alright, now, so to sum up everything, okay, the ovary, right, this ovary develops into fruit. Okay, the ovary wall, okay, becomes the pericarp, and pericarp, okay, consists of, you can see from here, this is the exocarp, and then we have the mesocarp, right, and then we have the endocarp, right, and then the triploid nucleus, like what I mentioned earlier, develops into endosperm. All right, this endosperm is the food storing tissue of the seeds that supplies nutrients to the embryo. Okay, so endosperm is for the developing embryo, actually. Right. Okay, next, right, the ovule. Okay, the ovule develops into seeds, which is inside the fruit. Ini fruit kan? Ovary jadi fruit. Right, so dalam fruit itu ada seeds kan? So the seeds it develop from ovule, right? So the ovule develops into seeds, which is inside the fruit. Now the zygote, okay, will develop into embryo. Can you see the embryo here? Right, which later, okay, the embryo will develop into sporophytes. Okay, so the embryo is packaged along with the endosperm into a seed. Can you see? A seed has the embryo, the endosperm, and the seed coat. Okay. Okay, now please take note that one flower, okay, has one ovary, which will develop into a fruit. So one flower has one ovary. So a flower has many ovules, okay, because one ovary has many ovules. So a flower may experience many double fertilization, meaning to say few pollen tubes are formed at a time inside a pistil. Okay, so a fruit has many seeds that came from many fertilized ovules. So you can see from here, this is one example of a flower, okay, which eventually forms an apple. Right, now this flower here, okay, it should be turned upside down. All right, so this will be the seed, this is the ovary which develop into the fruits. Okay, then here the bottom part of this fruit, since it's being upside down like this is a stigma and style that eventually degenerates. Okay, so this part of the fruit, all right, oops, connected to the plant by stalk. Okay, stalk, is it stalk? S-T-A-L-K. So okay, again you can see from here this papaya, okay, the stigma and style that degenerates. All right, and then these are the examples of many seeds. So these are many types of fruits, right? We have this accessory fruit, multiple fruit, aggregate fruit, and simple fruit. Okay, so this is not in your syllabus, so okay, so you just read it. Okay, so this diagram here, as in your module, it explains double fertilization process. Okay, and this video will show you the double fertilization process.
Flowering plants undergo a unique reproductive process where there are two fertilization events. This double fertilization event occurs between the male reproductive organ, the male gametophyte, and the female reproductive organ, the female gametophyte. Before the fertilization event can occur, the ovule has to undergo some changes. At present, the ovule contains one reproductive cell, known as the megaspore or mother cell. This cell is diploid and undergoes meiosis, producing 4 haploid megaspores. In the majority of species, 3 of these megaspores degenerate, leaving only one surviving megaspore. This surviving megaspore expands and undergoes 3 rounds of mitosis to produce 8 haploid nuclei. As the nuclei have not developed any individual division, they initially share the same cytoplasm. This complete structure is known as the embryo sac. Within the established embryo sac, cell walls begin to form between most of the nuclei. 3 cells named antipodal cells form opposite the opening of the ovule, known as the micropyle. Another 3 cells form above the micropyle. 2 of these are synergids, and the other is the egg cell. This leaves 2 nuclei in the center of the ovule. These central nuclei remain together in one large cell. It is the egg cell and this central nucleate cell which will eventually become part of the double fertilization event. In order for the double fertilization event to occur, the male gametes, the sperm, must travel from the anther to the embryo sac within the female reproductive organ. The pollen grain contains 2 main cells. A cell named the tube cell makes up the bulk of the pollen grain, and the sperm cell, which at this stage is known as the generative cell. To reach the embryo sac, a pollen grain must land on the stigma. Once landed, it begins to germinate. The tube cell forms a long structure down the style and into the ovary. The generative cell travels behind the tube cell nucleus. Once near the ovary, it divides by mitosis to produce 2 haploid sperm cells. The pollen tube reaches the micropyle and releases the sperm cells into the embryo sac. One of the 2 sperm cells fertilizes the egg cell. This produces a diploid zygote, which will become the embryo. The other sperm cell moves up and fuses with both of the central nuclei, forming a triploid cell. This unusual triploid cell develops into an endosperm and serves as the embryo's food supply during early development. It is only angiosperms, flowering plants, which have this double fertilization characteristic, where a diploid zygote and a triploid endosperm form. Gymnosperms, pines, tracheophytes, ferns, and non-tracheophytes, mosses, lack this double fertilization feature.
Okay, so that's about it. Okay, so see you in the next lesson.